some of the research that's being done now on neural functioning is that synapses aren't as straightforward as we thought they were. It looks here, and it looks certainly on our previous neurophysiology model, that a synapse is the union between a receptor, the gap then between that, and then the next cell. It's now known that synapses are actually, in the central nervous system anyway, what is known as tripartite. Tripartite. Three parts to the synapse. Now we see two. Well, what's the third one? Well, here's a very interesting little piece of information that's been developing now. It appears that astrocytes, and we've talked about astrocytes before, control a certain number of synapses. They have a little area of the central nervous system that's their own. Now, each astrocyte can control up to 140,000 synapses. There are two types of astrocytes. Astrocytes are glial cells, you might remember, which were once thought to just be the connective cells of the central nervous system, or of the nervous system. But astrocytes have two types. There are protoplasmic astrocytes and fibrous astrocytes. They do slightly different things. The protoplasmic astrocytes have many branches. They have processes that branch all over the place. And these envelop synapses. They literally cover and control a synapse. The fibrous astrocytes have many projections that don't branch, and they attach and envelop nodes of Ranvier. You saw the nodes of Ranvier before. They were the gaps in between the myelin sheaths where a saltatory conduction leaps from one to the next. Well, the fibrous astrocytes have projections that envelop the nodes of Ranvier. So if you can just envision here for a moment now, we've got an astrocyte with hundreds of feet or thousands of feet projecting out and covering the nodes of Ranvier of a whole range of different neurons. And then another astrocyte with projections coming out all over the place covering all of the synaptic connections. Remember, each neuron can communicate with up to 20,000 others. So you get how many projections there might be from an astrocyte.